Hey guys, I'm gonna go over chapter 13 lecture today. It's all about DNA. The first lesson is about identifying the substance of the gene. The second is the structure of DNA. And then the third is DNA replication. Oh, isn't that mouse cute? All right, so our objectives: identify the clues that bacterial transformation yielded about the gene. We're going to explain the role bacterial viruses played in identifying genetic material. We're going to describe the role of DNA in heredity. All right, so in the lesson one, we're going to talk about transformation, and this leads us to a scientist called Griffith. This is his experiment in a nutshell. So you will see petri dishes, all right, and we have this substance here, we have this substance here, right? We want to know why this substance ended up killing this mouse ultimately, right? And then when we injected this substance into this mouse, it didn't kill the mouse, right? Okay, so what we're looking at here is a Bunsen burner, a nice blue flame, nice and lit. So what they do is they put the test tube in that heat, and it potentially, and it should, as long as you do it the right amount of time, it will kill all of the bacteria or the contagion that is in that test tube. And we see that because, we know it was killed because it was given to the mouse and the mouse survived. Now, you can see here that this is a purple substance. What we're looking at here is a heat killed. We did put the heat killed in here, but it also had some of the live good bacteria, right? So if you have good live bacteria, but you still have the heat killed bad bacteria, and you put those two together to create this purple substance, what you're going to find out is that the bacteria in the good bacteria, DNA, will transform into bad because even though it was heat killed, there's still DNA floating around in that substance. And so ultimately transformation means that something that has got good DNA that's still living can transform into something bad. And that is why this poor little mouse is deceased now. It's very sad. So anyway, that is transformation in a nutshell. Oh, I should have been clicking that whole time. All right, so heat-killed disease-causing bacteria is the S type, and this is a harmless bacteria mixed together, and then you can't see down here, it does say that the bacteria, it says the mouse dies of pneumonia because of this. Okay, Avery's experiments. Scientists discovered that the nucleic acid DNA stores and transmits genetic information from one generation of bacteria to the next. So go ahead and read a little bit more about Avery, and we'll discuss those more in class. Bacterial viruses, or bacteriophages, are a kind of virus that infects bacteria. What you see here is the structure. They kind of look like a little lunar lander. They have DNA inside of this capsule here, or the head. They have tail fibers to help them attach to things, and they have a tail sheath that is very strong, and so is this head, this capsule. It's very good at resisting the body's macrophages. All right. So the Hershey and Chase experiment is the next one. What it talks about is bacteriophages with phosphorus 32 in their DNA, and bacteriophages with sulfur 35 in the protein coat that you can see down here. So this is Hershey-Chase experiment. Okay. So what ends up happening is that this phage infects bacterium. So what they did was they used these radioactive dyes, phosphorus 32, and they injected it inside of this part of the bacteriophage to see what would happen. And then they did the same here, but in this one here, it was on the protein coat. So it's only on the outside of the bacteriophage, because they were trying to figure out, like, what causes these bacteriophages to, harm organisms. They didn't understand how they injected themselves into cells and how they took over cells. So what we end up seeing, and they ended up learning that DNA, this was back before we didn't really understand DNA, they ended up learning that the bacteriophage inserts something that was in that capsule or that head into a bacterium. So they can see now that wherever the important substance was inside the capsule, and then in here there's no radioactivity, so they know that it didn't come from the protein coat, right? Because you can see all the radioactive substances around there. So here's the bacteriophage leaving after it injects and infects this, potentially. So now, after we had those few experiments, we now can look at DNA and understand it a little bit better, and we know that it is important. So the role of DNA. We know that it stores DNA. The DNA stores genetic information. For example, a flower may have genes for purple pigments. We know that it can copy the DNA. Every time a cell divides, it has to make an exact copy of that pigment for that gene, for example. And then we know that it expresses. So DNA puts genetic information to work. The cells use instructions from DNA to produce enzymes that make that purple pigment, for example. All right, so on to the structure of DNA. Look at those beautiful genetic variations on the wings of that butterfly. All right, so we're going to identify the chemical components of DNA. We're going to describe the clues that help scientists determine the structure. We're going to explain what double helix model shows about DNA. And here we go. I mentioned this earlier in the year when we did our pretest and when we were talking about our macromolecules. So DNA is made up of nucleotides joined into long strands or chains by covalent bonds. There's another vocab word that we learned in Chapter 2. So nucleic acids are made up of the building blocks called nucleotides. So this is a nucleotide you're looking at. All right, and nucleic acids were one of the 4 macromolecules, if you remember. That's very important. I told you it would be coming back and to keep remembering it. These are a little bit off, the arrows, but this is the base. This is your 5-carbon sugar, see the 5 sides? And then this is your phosphate group. So these 3 parts, you need to memorize them and know that they make up a nucleotide. All right, and so I hope that at one point in maybe 7th grade, you talked about the different nitrogenous bases. Adenine is A, guanine is G, cytosine is C, thymine is T. So here's one nucleotide. Here's another nucleotide. Oh, do I have a pen? I think I have a pen. All right, so here is a nucleotide. Oh, it's a little bit cut off. Whoops. All right. And then you have another nucleotide. And another. So how many nucleotides do you see here? There's 4, right? So this is one string of the ladder of DNA that you're seeing here. All right. So they will have covalent bonds between the nucleotides, what attaches the phosphate group to the sugar. All right. And so Chargaff's rule, A and T should be about equal in size, and C and G should be about equal in size. There's not a whole lot more than that than you need to know, but each nucleotide will make up 100%, right? So if I say that A is 20 and T is 20, and then that together makes 40, and then you'll have 30 for the other two nitrogenous bases. We'll go over that again a little bit more in class. Okay, so on to one of the most amazing women in science history. Her name is Rosalind Franklin. She used X-rays to study DNA, and she found out, she was the first person to say that DNA is a helix shape. So it's likely 2 strands is another conclusion she made. The nitrogenous bases are near the center of the molecule was another thing that she discovered. The sad part about it is that she didn't live long enough because the X-rays ended up giving her cancer. She didn't live long enough to win the Nobel Prize with these 2 guys. So this is Watson and Crick. They confirmed that DNA is a double helix in which two strands of nucleotide sequences are wound around one another, and we talked about the histones and everything like that, how they're wrapped around the histone proteins, right? So this is what a actual DNA double helix will look like, and they have anti-parallel strands. All that means is that the opposite base will be on the other side of the string, and they run in opposite directions. Okay, so the two strands in the DNA run in opposite directions. You have this blue part is representing your phosphate sugar backbone here. Phosphate sugar, phosphate sugar. So phosphate sugar, phosphate sugar, phosphate sugar in between each of these nitrogenous base pairs. Okay. So please remember that the double helix does have hydrogen bonding between the center of those C's and G's because it needs to be a weaker type of bond. So hydrogen bonds are weaker than covalent, and it needs to be weaker because of DNA replication. So we'll talk about that in the third lesson. So you can see here, guanine and cytosine, adenine, thymine, these are the base pairs. So you have your phosphate sugar, phosphate sugar, phosphate sugar backbone over there. All right. So it is your job to memorize the structure of this and what it looks like. And yes, this is supposed to be circling. Let's fix that. The formatting got messed up when I put it in Google Slides. It's supposed to be circling between here. Those are your hydrogen bonds. Okay. So the double helix base pairing, the two strands of DNA are held together by hydrogen bonds between the nitrogenous bases adenine and thymine, and between guanine and cytosine. Memorize those base pairs, guys. All right, last lesson, DNA replication. Look at this, 3D printing skulls. How cool is that? All right. We're going to explain the role of DNA polymerase in copying DNA. We're going to compare DNA replication in prokaryotic cells and eukaryotic cells. All right, so here's the DNA structure. Can you take a second and label all those for me? I don't think you can see what's happening down here. There we go. All right, DNA structure. Okay, so this is the sugar phosphate backbone. They made it the strands two different colors. And then you've got your nitrogenous bases here, and you've got the double helix is the overall structure, that swirly structure, okay? All right, so how do we copy DNA or replicate DNA? All right, so there is the direction of replication going this way and this way. What we call this right here is the replication fork, where it starts to split on both sides. Okay, because it's a eukaryotic cell we're looking at, they really do work very efficiently and in both directions. This right here is ATP synthase. It's helping to put everything back together. All right, so this is where the nucleotides are being added in this little ATP synthase bubble. All right, we'll talk more about that. The blue strand represents the original strand, and the new strand, okay, so this orange strand represents the new. So this orange strand is the new one being replicated. So every piece of DNA will always have one original strand and one new when going through replication. All right, so telomeres, they're the tips of eukaryotic chromosomes. The enzyme telomerase adds short repeated DNA sequences to telomeres as the chromosomes are replicated. Just memorize what this vocab word is. There shouldn't really be much more to it than that. All right, and then you can see the little dots. These little dots are telomeres at the end of the chromosomes. Okay, so prokaryotic DNA replication is quite simpler. There's a replication fork there and there. The new DNA is here, all right? It's a single circular strand of DNA, right? So it's going to happen much quicker. And here's the unreplicated DNA. We know prokaryotes reproduce really fast. So there's going to be you know, a lot of replication forks happening all at once. All right, so more replication forks here, here, and here. This would be a eukaryotic DNA replication. Obviously, we have more complicated DNA than a prokaryote, so we're going to have more replication forks, right? Oh, well, that is it. I will revisit some of these topics in class, and if you have any questions, please comment below the lecture, and make sure that you are reading through your textbook and reading through these images in your textbook for more practice, please. Bye!